today we are talking about fermentation. So, not all organisms carry out cell respiration. We talked. Cell respiration is also kind of, or at least the process we learned with oxidative phosphorylation, requires oxygen, but not all organisms need oxygen to carry out this process. Fermentation and anaerobic respiration enable cells to produce ATP without the use of oxygen. Ooh, that's not a word. Enable cells to produce ATP without the use of oxygen. The big difference between the two is that anaerobic respiration has an electron transport chain and fermentation does not. So based on what we know about the electron transport chain, which one of these do you think makes more ATP, anaerobic respiration or fermentation? Why? Yes, that electron transport chain is a ATP fest. We've got that ATP synthase that spins around and makes a bunch of ATP. We get most of our ATP in cell respiration from that electron transport chain. So fermentation isn't quite as efficient as anaerobic respiration, but it gets the job done. So, actually, make this really big. Fermentation is a way of harvesting chemical energy without oxygen or an electron transport chain. So fermentation harvests energy without oxygen or an electron transport chain. It is an extension of glycolysis that allows the continuous generation of ATP by substrate level phosphorylation. That's not how you spell phosphorylation. So, remember our ATP and citric acid cycle ATPs, or glycolysis and citric acid cycle ATPs, are put together by substrate level phosphorylation. But our electron transport chain generates ATP by oxidative phosphorylation. So, fermentation does not use oxidative phosphorylation at all. It only uses substrate level phosphorylation. But what we need, go away. In order to do this, we need sufficient NAD+. Because in glycolysis, we take NAD+ and we turn it into that electron carrier NADH. And in aerobic respiration, when we have that electron transport chain, we use that energy from that NADH. But without an electron transport chain, there's nothing to make NADH back into NAD+ for it to go back up to glycolysis. Do you guys get where I'm coming from? Yeah? Okay. So, fermentation is glycolysis plus the reactions that regenerate NAD+. And that happens by transferring electrons from NADH to pyruvate, which is the end product of glycolysis, or a pyruvate derivative. So, glycolysis is kind of a universal pathway. Fermentation is glycolysis plus the regeneration of NAD+. Just like the cellular respiration that we talked about was glycolysis plus the oxidation of pyruvate plus the citric acid cycle plus the electron transport chain. So, fermentation is glycolysis plus this regeneration of NAD+. There are two types of fermentation. Two types of fermentation. The first is alcohol fermentation. In alcohol fermentation, we turn pyruvate into ethanol or ethyl alcohol. In 2 steps. First, we release carbon dioxide from pyruvate, release CO2 from pyruvate, this creates acetaldehyde. Acetaldehyde. That's step 1. We just take that CO2 just like we did in oxidating pyruvate, but just a kind of a different thing. We're releasing CO2. And once we have that acetaldehyde, we take it. It's actually a 2-carbon acetaldehyde. And we reduce it to ethanol by NADH. This yields NAD+. So instead of taking the pyruvate and the NADH and sending it to the citric acid cycle and the electron transport chain, we take it, we release a carbon dioxide from pyruvate, we reduce that with NADH, and we get ethanol and NAD+. The ethanol is a byproduct, and the NADH goes back up to glycolysis so we can get more ATP. So if we're going to diagram that, we start with 2 ADP plus our inorganic phosphate. And through the process of glycolysis, we get 2 ATP. Like I said, this is glycolysis. Glycolysis yields pyruvate. And NADH, or 2-NADH. That pyruvate releases CO2 and gives us 2 acetaldehydes. Those NADHs and acetaldehydes go through the process of NAD+ regeneration. Actually, that will give us 2 ethanol and 2 NAD+, and those NAD+ go into glycolysis. So if we take, we'll make it a little bigger. We got our 2 ADP. We're making ATP through glycolysis. And after we've got the ATP, we have pyruvate and NADH. That pyruvate and NADH turn into 2 acetaldehydes. Those acetaldehydes and those NADHs go into NAD+ regeneration. We get 2 ethanol and NAD+. I feel like I'm missing something. Nope. All right. This is the process that happens. Sorry. When we make alcoholic beverages, wine, beer, all those things are made through this process. So that's alcoholic fermentation. Our bodies don't do this. Our bodies, whenever we don't have oxygen, do something called lactic acid fermentation. Where are we at? Lactic acid fermentation. In this type of fermentation, pyruvate is reduced directly. By NADH to form lactate, which is just an ionized form of lactic acid. So instead of having that like halfway acetaldehyde intermediate, we just directly reduce pyruvate. This regenerates NAD plus without releasing carbon dioxide. This regenerates NAD plus without releasing carbon dioxide. This process is what happens when we make cheeses and yogurts. We have certain bacteria that we use to ferment milk through lactic acid fermentation. That's why yogurt tastes the way that it does. This is also what happens in human muscle cells. When ATP is scarce. So if you've ever sprinted down a track or across the hallway, right? It gets to the point where your muscles are just burning, right? Like it feels like you cannot physically move. And when you stop moving, your muscles just kind of tense up, right? That's why when people like roll out their muscles, not only are they loosening it, but they're also kind of distributing that lactic acid so it breaks down a lot faster. That lactic acid fermentation is what burns. Because you've used up all the ATP you have, so your cells go into emergency mode, and that emergency mode is lactic acid fermentation. So it just keeps going through glycolysis because you don't have enough oxygen, because you're not breathing enough to exert the energy that you are. So when we diagram this one, I'll try to make this one a little neater because I feel like the last one got messy. We have our 2 ATP, or ADP, sorry, and our inorganic phosphates turning into ATP. This is through glycolysis. This yields pyruvate, as we know and love it. Also NADH. That NADH goes into NAD plus regeneration. And that pyruvate gets turned into 2-lactate. And like I said, that lactate is just an ionized form of lactic acid. If you want to put lactic acid there, it probably wouldn't hurt. This gives us our NAD plus back, and that NAD plus can go back and do its job in glycolysis. So that's fermentation. Now, first of all, do I have any questions? We are going to compare fermentation aerobic respiration, which is what we learned about all last week, and anaerobic respiration. Anaerobic respiration still has an electron transport chain. It just uses some other final electron acceptor besides oxygen. So it'll use like a sulfur compound or something of that nature to be that last kind of stand-in where oxygen would be. So all three use glycolysis and NAD+. That NAD+ is an oxidizing agent. That accepts electrons from whatever it is we're eating. We learned yesterday that glycolysis isn't just breaking down glucose, that proteins, other carbohydrates, and fats also get fed into the process at some point. The key difference is how we get from NADH to NAD+. That's our key difference. They all start out the same. So like we said, in fermentation, our electron acceptor is pyruvate. In lactic acid fermentation, or acetaldehyde in alcohol fermentation. In either of the respirations, we have our electron transport chain and that is how we get from NADH to NAD+. Another key difference, another one, is the amount of ATP. Respiration makes way, way, way more than fermentation. So more complicated organisms tend to carry out cell respiration, where fermentation is usually done by smaller organisms that don't require a heck of a lot of energy. So, speaking of those organisms, there's two types we're going to talk about. There's a lot of these, like a lot of these, and if you ever take a microbiology course, you will learn them all. But we're going to talk about different types of organisms that carry out these processes. The first one is an obligate anaerobe. Obligate anaerobe. What do you think an obligate anaerobe is going to carry out? So, an obligate anaerobe can only do fermentation and anaerobic respiration. This is because they can't survive in the presence of oxygen. Can't survive in the presence of oxygen. This is stuff like in the unoxygenated parts of the world. Really, really low levels of oxygen. Dissolved in water with little to no oxygen content, right? Suspended in media that hasn't been exposed to the air. So we've got obligate anaerobes, but we also have facultative anaerobes. This is like yeast and bacteria. These can use fermentation or respiration. So if they're in an environment with a lot of oxygen, they can carry out respiration just like us. But if they're in an area with no oxygen, they can carry out fermentation or anaerobic respiration. But there is a caveat here. To make the same amount of ATP, they have to consume sugar at a much, much higher rate while fermenting. So if they're in an environment with no oxygen but have plenty of sugars available, then they'll be fine. They can make the same amount of ATP as they would regardless. Any questions about this? Yeah, one last kind of connection to draw. Okay. And that is evolution and glycolysis. Evolution and glycolysis. So, throughout the course of the year, we've talked about evolution in a couple of different ways. We mentioned endosymbiotic theory. Evolution is just kind of one of those key terms that drives everything home. So, ancient prokaryotes, that's not how you spell ancient. Ancient prokaryotes are thought to have used glycolysis before there was oxygen in the atmosphere. That's kind of messy, but we have found bacteria dating as far back as 3.5 billion years ago. But we know that oxygen in the atmosphere only appeared about 2.7 billion years ago. Which means these bacteria had to get energy somehow. before there was oxygen available. Therefore, if you ever see these three dots, I'm not sure which of my shorthandy things I've passed on to you, but those three dots mean therefore because people get lazy. Myself included, I don't want to write out therefore. They're early prokaryotes exclusively use glycolysis. We think this is true because glycolysis is the most widespread metabolic pathway. On Earth. Which means it evolved early. Like real early. Oh no. Also, it happens in the cytosol. We talked about how we don't enter the mitochondria until we are pyruvate. Because glycolysis happened in the cytosol, it means it does not require membrane-bound organelles. Which is likely because this happened before our early eukaryote ancestor sucked up a prokaryote ancestor and made mitochondria. How are we doing? Any questions? All right. There is an exit ticket on Canvas. If you could fill that out for me, please.